Hello everyone, today we'll talk about the search for the genetic material. The very first scientist named was Mister. He discovered nuclein as the genetic material because this particular structure is found in the nucleus. That is why he named this chemical compound as nuclein. After the mixture, there were many scientists who conducted a series of experiments and began the journey for the search of genetic material. The very first experiment conducted by a scientist known as Frederick Griffith. He conducted a set of experiments in 1928. He called these set of experiments as transforming principle. What he did, he took a bacteria Streptococcus pneumonia and he told us that there are two strains of these bacteria. This is a bacteria which causes pneumonia in certain organisms. So he told us there are two strains of this bacteria. The first strain is called R strain and other strain is called S strain of this bacteria. R strain, these are the set of bacteria which were producing rough colonies in the culture plate, and these are without capsules. These are not enclosed by any capsule. S strain bacteria was producing smooth colonies on the culture plate and these have the capsules around it. If we talk about these two type of strain, the R strain is non-toxic, or we call it as non-virulent, and the S strain is highly virulent form of this bacteria. Now, what was the exact experiment? So, he took a mice, he injected the S strain bacteria into the mice, and what happened? The mice died. The second observation was, he took a mice, he injected the R strain bacteria into the mice and the mice lived. The third observation is, he took the mice again, he injected the heat killed S strain into the bacteria, and what happened here? The mice lived. Now the fourth observation he observed is, he injected the mice with the heat killed S strain along with the R strain. Now what happened here? The mice died. This fourth observation was the eureka moment for the Griffith. Why? Because earlier the heat killed S strain was not killing the bacteria, was not killing the mice, and R strain also was not killing the mice. But in the fourth observation, the mice died. So what was the conclusion of this experiment? That somehow the R strain bacteria were transformed by the S strain, but he was unable to understand the cause of this transformation. He said that somehow this R strain bacteria got converted into the virulent form by the S strain, and he called this as transforming principle. This is the reason behind the R strain bacteria got converted into the S strain virulent form. This is called as transforming principle. So I hope you understand this experiment, and this experiment is very much clear to you. If you have any questions, you can leave the comment. Thank you so much for today and have a great day.